Okay, folks, I'm going to go back and actually put in part two. Somehow we lost part two of our video. So away we go here. Getting to this slide, we look at the history of the Earth in terms of the rise of oxygen in the atmosphere. When the Earth first formed, what you have to realize is there wasn't anything that was producing oxygen. Now, there weren't any bacteria that were producing it. There were no plants that were producing it. So really, how did it get here? Well, first of all, you have to look at the first life forms. And the first life forms that arrived on the planet that we have any evidence of are prokaryotic cells. So prokaryotic cells comes from an interesting little word, pro, which means before there was, and karyotic, which means having a nucleus. So it literally means before there was a nucleus. Now there is evidence that goes back quite a bit in terms of what we're talking about here with these little sort of microfossils. So I'm going to pop out to that and show you, and I've got it stored here. Here we go. These little things called stromatolites. You find them just offshore. You can find these in Australia, and they're, let's see here. Get the picture to come up. Just one moment. Thank you, Wikipedia. And these interesting little sort of green, they almost look like mushroom caps. These things are what we call stromatolites, and they're layer upon layer upon layer of bacteria. Now, what you have to understand with something like that, bacteria, as they grow in the layers, would grow closer towards the sunlight. And as layer upon layer was added, the ancient layers that go down as far as you can imagine in the middle of that stromatolite that you see there represent the oldest colonies. So it's when you look at stromatolites, evidence of them can be found quite readily. Oops, there you go. And they have this interesting sort of wavy appearance, these wavy lines. So you're sort of looking at a cross section of a stromatolite mat. Go back and give you an even better view of a stromatolite. If you look at a stromatolite underwater, you would be looking at this. And, oops, I just want to see the picture, thank you. That's what they look like underwater. So you only really find them in very shallow water, and that's so that they can photosynthesize effectively. But they leave behind a very ancient tale of what was happening in early life. And this is a neat picture because you can see the wavy appearance of the stromatolite, which is what we find in the fossil records. And then if you look at the image and you see the little green dots in that little sort of circle to the right, those are the stromatolite individual sort of bacterial cells. These are like blue-green algae. They're a cousin to that. And that's what we're talking about. Now, the rise in oxygen in Earth's atmosphere, here's a neat graphic. We know about 2 billion years ago that photosynthetic bacterial cells got going. And if you look at the rise of that red line, you can see the oxygen, the atmospheric oxygen, absolutely rise, and then it steadies out. So we kind of hit a saturation point, a certain amount of oxygen in the atmosphere. Now, oxygen itself was incredibly important to the formation of the planet, planetary life, because oxygen helps form ozone. Now, you might have heard of that before. It's an interesting combination of oxygen atoms. Instead of O2, ozone is O3. You find it in the upper atmosphere. Go up very high. Go to the stratosphere. And UV light helps oxygen molecules stick together not as O2, but actually as an interesting grouping of O3. The UV light helps them to stick together. And you might remember in the news several years ago, there were holes in the ozone layer because we were using these chemicals. They were mostly refrigerants, known as one particular one was really nasty, chlorofluorocarbons, or CFCs. And they were making it so that the oxygen molecules, the O3, couldn't stick together. And without those oxygen molecules sticking together, there was nothing that was using the UV light to hang out. And that UV radiation was coming down to the planet and giving everybody a really bad suntan and, well, worse, it was worsening skin conditions such as melanomas, also known as skin cancer. So the rise of oxygen in the atmosphere, you have to understand, oxygen is pretty toxic to a lot of bacteria. The archaeobacteria couldn't stand it. To them, it was like a metabolic poison. When you look at what oxygen does, um, and then we'll go to, I'll just call this one up on the spot, oxygen bands, oxygen bands, and I should be able to get it if I type iron. If you go look in the Earth's crust, you can see, if you dig down, where the iron bands begin to form. Oops. And it looks about like this. So iron ore, in the presence of oxygen, it will oxidize and form this sort of rust-like color. If you go down even further, you don't even see these rust bands. This is how we know that oxygen was accumulating in the Earth 2.2 billion years ago. We just figure out the age of the rock. We go very deep and we figure out the age of the sample, and we get an idea of when oxygen was being produced on our planet. Stromatolites are a tremendous clue. So don't forget about them, okay? Because I won't. Turned out that oxygen was useful for the critters that came along next. For example, plants use oxygen, they burn their own fuel, right? Mitochondria, those little bacteria you know about, or you might have heard about. These little critters, these little bacteria could use oxygen and they could increase their metabolic rate. You and I can't survive without it. So through evolution, something came up, that a critter that was able to use oxygen as part of its metabolism. And really, we never looked back. It's only the archaeobacteria that try to hide from oxygen down deep in the ocean in geothermal vents where there's very little oxygen, right? Or in these deep, dark caverns where you find very little oxygen in these horribly ancient Hadean conditions, right? When the Earth first formed. There is sort of a little bit of thought you have to put into, where did cells with a nucleus come from? So the modern cell, let's just draw an animal cell, has a cell membrane and has a nucleus. Kind of looks like this, and a little dark pit in the middle like that. And then it has these little organelles that run throughout, these little ribosomes and all this kind of stuff. And it has an interesting little energy producer in there, this little guy, called a mitochondrion. Now this stumped everybody for a long time, because your mitochondria produce energy. Think of them as gas-powered generators in your cells. Okay? And generators run on fuel. Without getting too much, I think, into the grade 12 aspect of looking at cellular biology, these little generators, little energy generators in our cells, if you examine them closely enough, they're very interesting, because it turns out that they're bacteria. We can pull them out of your cells and we find that they have their own DNA. They're prokaryotic because they don't have a nucleus. We can injure them with antibiotics. So what the heck is going on with this? Why are mitochondria basically aerobic bacteria? I'll make a little note of that. These are aerobic bacteria, which means they love oxygen and they make energy in the presence of it. What's up with that? Well, let me explain. Move along here. There's a theory called the endosymbiotic theory. Yes, you have to know it. It's absolutely critical. Endo means inside, like you have an exoskeleton. So inside us, there is, and let's switch the color because I'm getting tired of that color. There's a symbiote. A symbiote is an organism that lives in association with another one, with another. Now, you've probably seen the sharks and the remora. If you've ever looked at a lichen, that's algae living with fungi. So sometimes we have these deals where two critters live together. On the African safari, you'll see the birds keeping the giraffes clean, they'll keep the rhinoceros clean. That's sort of a symbiosis, a mutualism. You help me, I help you. But wait a minute. When you think about this, you never saw the bird on the giraffe live inside the giraffe. I mean, that would seem absolutely bizarre. But there are some things that come to live so close together that one actually lives inside the other one, and they just form a permanent sort of residence. So when we look at the rise of the eukaryotic cells here, what we think is that a modern cell is actually a composite. It's more than just one thing. We think that the cell arose. Let's go to a blue here. We think that the cell, and I'll draw it like a Pac-Man, one day went out seeking or engulfing another food source. But what actually happened, it engulfed a food source. Now, this is just a theory, mind you, but we have quite a bit of evidence because they live together now. And if you look at this up close, I'll zoom in. So here's our little, this is generally how you'll see a mitochondrion drawn. It's got these neat inner membrane folds that go back and forth like this. We think that what happened was that it was engulfed, and I'll just do a little eraser-rooney. Oops. I think I can get rid of it now. Oh no, it's not going to let me. So we'll draw before and after, shall we? Okay, we think that engulfed it, and then what happened was that it realized what it had. In fact, it was a very good deal. When they came to live together, and we know this happened because mitochondria are still with us, and our cells have never digested and eaten them. In fact, it's like they adopted them. And I think there's a good reason for that. They got a very good deal. These guys came to live inside of us, and if you feed them a little bit of your food, they can produce all the energy the cell could imagine. Well, not all that it could imagine, but way more than the cell ever made. Some evidence for the endosymbiotic theory when you look closely at the cell. This little guy, when he came in, when he was swallowed, generally what happens when something is brought in through our cell membrane, which is right here, and I'll just put a little note here, cell membrane. When something gets brought into our cell membrane, it kind of gets coated by it. So this thing not only had its membrane, its black outline, but when you look at these little guys now, what you'll notice is they've got an extra layer around the outside. It's like they've got extra bagging. And you have to ask yourself a question, why do these things have more than one set of membranes? The evidence for the endosymbiotic theory, I mean, remember, when I say theory, I'm not talking about a hunch. This deal went down, but we just went around to see it. Because this guy has his own DNA, has his own membrane, has his own cytoplasm, which has its own name, but we'll talk about that in grade 12, and produces a tremendous amount of energy for the cell. Really, all you have to feed this thing is glucose, and it will crank out the glucose is basically the gas that these little generators run on, and then they produce the real fuel that cells need. Now I might as well just tell you what it is. You feed them glucose, and what they crank out is, think of it as the electricity. These guys crank out ATP, which is a high energy molecule known as adenosine triphosphate. Your cells actually run on that. Glucose is just like the gas. A generator might produce electricity, if you think of that as really what keeps your house going. In this case, what keeps a cell going is actually ATP. So I'm giving you a little bit of advanced information. It's okay. You can bring me a coffee any old time you feel like. Um, Two cream, Tim Hortons. It's up to you. All right. But all seriousness here, folks, this is what we think happened. That sort of there was a deal that basically happened between two cells. And I really don't like the way they've shown this because this cell here, they call it a primitive, sort of a primitive cell. You could call this cell what we call the eukaryote. It was kind of getting around to organizing its nucleus. So I think this is just annoying. Okay. And the reason I say that is it's putting its DNA kind of inside of a nuclear envelope. So maybe the cell's evolving. It's getting closer to being a true nucleated cell, close to being a eukaryote. It wasn't quite there, so you can call it an eukaryote for now. What happened though is you'll see that the this guy came in and he got coated with an extra dose of the membrane and came to live, an active life here. It's a good deal. The mitochondrion gets fed, gets a home, gets protected. Man, this is a great deal. And you see these kind of relationships all the time. Shark and remora, same kind of thing. Remora gets to eat. I don't think too much is going to come after the remora when the shark is hanging around, okay? You get the idea. The remora hangs off the shark. It doesn't have too many predators. That's a good deal for the remora. Good deal for the shark. The remora keeps it clean. We think that this deal happened more than once. In fact, we know it because if you look at a plant cell, um, if you had a primitive eukaryote that engulfed chloroplasts, now this is an even more amazing deal. If this guy right here produces energy for the cell, then what produced the gas for the generator? Think about it that. You've got to have gas to run your generator, and then the generator will produce all the power you want. Oh, how about a solar powered gas maker? Because that's what you got here. When you look at a chloroplast, see those green bacteria right there? In fact, when we look at a chloroplast, it is trapped photosynthetic bacteria. It's the same thing. It's got its own DNA. It resembles other bacteria that photosynthesize, and it's highly efficient at what it does. So here you go, photosynthetic bacteria. So we had deal number one and deal number two. Here's the kicker. Animal cells appear to have only made a deal with the aerobic bacteria here. Okay, so this would be an animal cell scenario. But then this one came along. Then we got a plant cell scenario. How do we know this happened? Well, folks, if you look closely at a chloroplast with all the evidence and everything it's telling you, I'm going to throw in one extra little bit. Guess what it's coated in? That's right, ladies and gentlemen. As it came in, it got coated by the membrane of its engulfing host. So a theory was brought up to say, oh, these symbiotes came to live within modern cells, and we call it the endosymbiotic theory. Now we know that fungi never made this deal, right? Not the same way plants did, but fungi do have fungus, like mushrooms, toadstools, things like that. They have mitochondria too. So if you go back far enough, maybe fungi didn't make a deal for like plants did. They didn't seize that opportunity. But when you think about the primitive eukaryotic cell, this could become all kinds of things. Animal cells, those ones called protists, which are going to come up later in the course. Fungi. The list goes on and on, folks. The list goes on and on. So when they talked 2 billion years ago, prokaryotic cells developing internal membranes, let's change the color just so I can give you a better visual. When you look at a chloroplast, what we know with some of their some of chloroplast's relatives, they started to stack like this, these little photosynthetic layers. Think of them as like little solar panels. If you look at a modern chloroplast, okay, this would be like an older version. If you look at a modern chloroplast, you will find these beautiful photosynthetic stacks of they look like pancakes. And a chloroplast is extremely advanced. These things are connected like this, and the chloroplasts in plants are just amazingly complex, and they do exactly what they want, which is to photosynthesize. So it was the development of this pigment into little layers, because what you wanted to do was, in the presence of sunlight, some of these critters learned how to become solar powered. So they were able to take solar energy and use it to make a fuel, which we all know as glucose. It just turns out that, just like the evolution of a solar panel, these guys evolved incredible complexity to get as much solar energy as they could to crank out exactly what plants need to live off, which is glucose. And if you like maple syrup, ladies and gentlemen, you can thank these chloroplasts, because they're the ones that made it in plants. So we'll do a happy dance. Yay! These guys were also producing glucose. But this is the old sort of primitive, think of it as a really early chloroplast. Okay? And those kind of bacteria are still around. Blue-green algae that float around like kind of foamy scum on Clucas Lake. You might have seen it. These guys here, they also produce glucose, or produce sugars. So we know that bacteria can do it. So when you heard that the first photosynthetic organisms on the planet were plants, you heard wrong. It's actually been bacteria. 2.2 billion years ago, newsflash, there weren't any plants. So here's a little sort of overview of it, right? The aerobic bacteria, aerobic meaning they used oxygen, right? These kind of bacteria loved oxygen and they could make energy with it. You get the idea. The little snack session went down, and there you go. They mentioned ATP here, as I did earlier, which is the fuel that cells actually run on. It's not that early cells couldn't make their own chemical energy, their own ATP. It's just that when they made a deal with this guy, and that was like nitro, that little guy popped out all the ATP you could even imagine. In fact, it's very good at cranking out about 36 ATP for basically a small amount of glucose. Without getting, I don't want to get into basically that biochemical cycle, but these guys can crank it out. And these guys, think of it as every cycle, these guys can crank out about 36 or 38 ATP. Before, cells were only cranking out about 2 ATPs for every glucose molecule. And it was just such a good deal that, well, the cell decided not to consume this symbiote. In fact, that's what made it an endosymbiote. Here's the deal we've already talked about sort of ad nauseum. The engulfing of a chloroplast by probably the early sort of plant-like, going to be plant-like cell. Neat stuff. And yes, chloroplasts, if they're removed from plants, here's more evidence. Plants can't grow them. The chloroplasts themselves, they just divide asexually in the plant. They just keep dividing and dividing. But if you remove them from a plant, plants can't produce them, because there is no DNA in here to make these things. That's because these little critters are their own organism. So at this point, they sort of raise a little sidebar here about sexual reproduction and multicellularity. Now, if you ever watch bacteria, if you leave bacteria out, they'll divide like crazy. It'll start as like one little cell, and then you've got two, and then you've got four, six, eight, and this is all like the worst part of an infection. Now, the only reason bacteria will decide to reproduce is if there's sort of an opportunity. So, asexual reproduction is when you produce clones of yourself. So, let's write that in. That's cloning, right? Spider plants do this. If you've seen them, they'll have a little runner, right? Plants don't have to have sex to produce another plant. They can just grow a clone over yonder, if you will. The daughter cells will be exact copies of the parent cell. So that is, you have to take note of that because that makes no diversity, right? Imagine just cloning yourself to reproduce. Nothing new. That is really bad because what happens is you're restricting genetic variation. I mean, really, what would happen if one of those bacteria that cloned itself was very easily killed by one variety of antibiotic? Now we've got a whole army that has the same problem. You're not going to see much variation going on in that population. So, when is it that sexual reproduction happens? Now, this is going to seem a little strange. It's a little hard to figure it out. Sexual reproduction is when you shuffle the deck of genetic cards. Okay? So think of your DNA, your chromosomes as being cards in a deck. If you don't have sexual reproduction, right, the offspring themselves will just look exactly like the parents. They'll have their characteristics. Well, they'll come from the one parent. That's not good. Genetically, there's no change. So when times get tough, bacteria will have sex, and they're doing sexual reproduction so that they can produce variations of offspring. That gives their offspring a better chance, maybe, just maybe, increased probability, that you'll get a new, sort of stronger bacterium. It's like when we have children, right? We're hoping that the next generation will be greater than ourselves. So when a child is produced by the union of a sperm and an egg, it's a new genetic combination. So it's got a chance in the new world. Maybe that's the right combination. Maybe that's the new hope. Sorry, I have to borrow a cheesy Star Wars line. This this is the whole reason for sexual reproduction is to produce change for a changing world. But I'll tell you right now, if bacteria encounter a food source and times are good, if you leave spaghetti sauce out on the stove, hey, times are good. They don't, the environment is ripe. They're not going to slow down and have sex. There's a tremendous opportunity here. And bacteria will just asexually clone themselves, right? But if you look in hospitals where a lot of antibiotics and things are being used, bacteria, you might have heard of bacteria that are resistant to a lot of antibiotics. Well, those bacteria are having sex and they're shuffling their genetic decks, trying to come up with that combination, that special genetic version that can resist these antibiotics we're using against them. So, sexual reproduction, ladies and gentlemen, is all about, I'm going to make a little side note, is all about increasing diversity. It's a shuffle. That doesn't mean there's new genes. It just means that they're trying combinations. So before I leave this, what I should say is that if you think of a deck of cards, think of mutation as being sort of a new card in the deck. And then think of sex as in shuffling the cards to try to get a favorable combination. If this turns out to be a brand new card, maybe it's I'll hit undo. Let's say maybe what you have is you got the Joker, and it's Joker's Wild. Hey, if you stick another Joker into the card game, right, and Jokers are wild, ooh, that could be an advantage in the card game. The analogy that I'm trying to make here is mutation can over time drive new combinations. And those new combinations are basically like genes being added, right? New DNA combos, like a Joker's wild card into a card game. Now, it's not that all mutations are good. In fact, a lot of mutations are really bad, and the organisms that produce them just die a silent death and they're never to be seen again. Okay? Here's an example of a mutation that really doesn't work out in nature. When you look at albinism, I mean, it's a mutation that makes a lot of organisms, they're a bit weaker, a bit more sickly, they're not as hardy, and it makes them harder for them to reproduce and survive into the next generation. Albino moose, for example, they really don't live a long life and they're not as likely to pass along that gene. That is not a positive mutation. That doesn't improve the survivability of the critter. However, if you got a mutation that gave you slightly better vision in the dark, made you a better hunter, hey, that would keep you fed and that could give you an advantage while hunting. So moving forward, that gene would get passed on to your offspring. So in a way, these mutations are the driving force for change in evolution. And the reason the mutations happen is just when we're copying genetic material, sometimes mistakes happen. Your cells aren't perfect copiers. And what happens is you can get genes kind of being rewritten almost by accident. You've heard of mutations. Sometimes your cells will mutate in the presence of sunlight and you can get skin cancer. That's not so good, right? So there's a nice example of a mutation being a very negative thing. Okay, I could go on and on, folks, but that's for another time and another lecture. So that ends 17.2, the missing half of the movie. What I'll do is I'll stitch this together with the first half of 17.2 and upload it. So, ladies and gentlemen, it's been Mr. B. You folks have a good one. And I got to get out of iOS 7 here. It's kind of new. All right. Thanks, folks.